Imagine two patients sitting in an oncology clinic. They're the exact same age, they have identical medical histories, and they both received the exact same terrifying diagnosis.
Yeah, triple-negative breast cancer.
Yeah.
Or TNBC.
Right. And they both undergo the standard, really grueling protocol, which is neoadjuvant chemotherapy. But then, 6 months later, the surgeon operates.
And this is where it gets crazy.
Exactly. Patient A has achieved a pathological complete response, so no active cancer cells remain at all. But patient B, despite enduring the exact same toxicity from the chemo, still has aggressive residual disease.
It's just brutal. And, for decades, that divergence has been one of the most frustrating black boxes in all of oncology.
It really is. So for you listening, that is our mission today. We are doing a deep dive into a massive stack of data that finally cracks that black box wide open.
Oh, it completely blows the doors off. We're talking about a landmark 2026 Nature paper. They tracked over 400,000 individual cells from 101 patients.
Which is an insane amount of data. But before we get into the biology, let's just clearly define the terms for everyone. What exactly makes TNBC so tricky?
Well, the triple negative part means the cancer lacks the 3 standard receptors. It doesn't have estrogen, progesterone, or HER2 receptors.
Right, so you can't use those targeted hormone therapies.
Exactly. You're left relying heavily on the sheer systemic toxicity of neoadjuvant chemotherapy.
And neoadjuvant just means giving the chemo before the surgery, like to try and shrink the tumor first?
Spot on. But why that blunt weapon works miracles in one body and, completely misfires in another, that's a question of microscopic cellular identity. We just didn't have the tools to see it before.
Right, which brings us to the tech. The reason we can finally solve this is because of a huge leap in spatial and transcriptomic technology. We moved from old-school bulk sequencing to single-cell RNA sequencing.
Yeah, combined with spatial transcriptomics, it changes everything. I mean, historically, with bulk sequencing, we'd take a tissue biopsy, extract all the RNA, and sequence it together.
Which is basically the classic city in a blender analogy.
Oh, I love that analogy.
Yeah, it's like you take an entire metropolitan area, right? You throw it into a giant blender, pour out the smoothie, and then try to guess the city's demographics based on the chemical makeup.
Right. You measure high sugar, so okay, there are bakeries in there somewhere.
Exactly. But you have absolutely no idea if those bakeries are next to the schools or if they're miles away in the industrial district. You lose all the structural context.
And in cancer biology, context is literal survival. So single-cell RNA sequencing, or cRNA-seq, fixes that. It uses these microfluidics to capture individual cells in tiny lipid droplets.
Wow.
Yeah, along with uniquely bar-coded beads. So we get a distinct genetic profile for every single cell. But this 2026 study went even further. They layered on spatial transcriptomics using Xenium and Visium platforms.
So they're using fluorescent probes that bind to specific RNA right there on the intact tissue slide.
Precisely. So we upgrade from a blended smoothie to, essentially Google Earth layered with a super invasive census.
We know the molecular identity of every cell, what it's doing, and exactly what street it lives on inside the tumor.
Right. And when they applied this mapping to those 400,000 cells, they started by just isolating the cancer cells. They wanted to see the big patient-level patterns.
And they ran these unsupervised clustering algorithms and found 4 distinct archetypes, right?
Yeah, 4 main gangs running the tumors. Archetype 1, or ARC1, is luminal secretory-like. It actually retains some genetic memory of normal milk-secreting breast tissue.
Oh, interesting.
But it shows high rates of structural breakdown. And then we have ARC2, which is basal-like.
And looking at the clinical outcomes, ARC2 is the undisputed villain here.
Oh, without a doubt. It is heavily associated with residual disease. If a patient has an ARC2-dominated tumor, standard chemotherapy is almost certainly going to fail.
Because the data shows these ARC2 cells heavily express a gene called mTDH, right?
Yes, metadherin. It's a known driver of chemoresistance. It basically interacts with survival pathways to suppress stress. So the cell just shrugs off the DNA damage from the chemo.
That's terrifying. But on the flip side, we have ARC3. This is the interferon responsive archetype.
The hero of our story, relatively speaking.
Right, because it's highly associated with a pathological complete response. If you have ARC3, the chemotherapy works beautifully. But wait, looking at the methodology here, I actually have to challenge this finding.
Oh, go for it.
Well, interferon signaling is a classic immune system pathway, right? It's how our immune cells sound the alarm.
That's true.
So in the old bulk sequencing days, an interferon signature just meant you had a lot of immune cells trapped in the tumor smoothie. How do they know this signal is actually coming from the cancer cells themselves?
That is exactly the skepticism the researchers expected. But because they used single-cell resolution, they computationally isolated cells using epithelial markers, like EPSAM.
Oh, so they completely filtered out all the actual immune cells before running the numbers.
Exactly. The data proved unequivocally that the malignant cancer cells are upregulating these interferon pathways themselves. The cancer is intrinsically driving an immune signal.
Wow, so the tumor is mimicking the immune system. That is a massive paradigm shift.
It really is. Oh, and just to round out the four, ARC4 is androgen receptor enriched, so it relies on completely different pathways driven by male hormones.
Right. And researchers actually use a mnemonic for these, right? The ABIA mnemonic.
Yes, ABIA. A for apoptotic or luminal in ARC1, B for basal, which means bad outcomes in ARC2, I for interferon, which means ideal outcomes in ARC3, and A for androgen in ARC4.
That's super helpful. Okay, so let's do a quick recap for you listening. Bullet 1. We zoomed in and found 4 main tumor archetypes. Bullet 2. We know the basal ARC2 resists treatment while the immune-mimicking ARC3 is highly vulnerable.
Right.
So here's a question for you to think about. If you could magically peek into a biopsy, which of these cellular gangs would you hope to see running the show? Obviously, the ARC3 interferon gang.
Definitely. But knowing the broad archetype isn't enough. We have to drill down deeper. Not every single cell in an ARC3 tumor is doing the exact same thing.
Right, we're getting into intra-tumoral heterogeneity. We need to know what the individual gang members are doing on a daily basis.
Exactly. And to do that, the researchers used some intense math, specifically an algorithm called non-negative matrix factorization, or NMF.
NMF.
Yeah, basically extracts these distinct patterns of gene expression, which they called metaprograms. They found 13 of them.
And a metaprogram is basically like an app running on a smartphone, right? It's a network of genes working together to do one specific job.
That is a perfect way to describe it. And out of those 13 apps, they found 4 that are strictly linked to a really good response to chemotherapy.
Okay, let's break those 4 down. The first is M7S, and the S stands for S phase, right?
Yeah, the part of the cell cycle where the cell is actively unwinding and copying its DNA.
Got it. And the second is M13ER stress, meaning the cell's endoplasmic reticulum, its protein folding factory, is under massive stress.
Right. And the mechanics of why dividing and stressing are actually good things here is crucial.
Yeah, explain that, because you'd think a rapidly dividing cancer cell is bad news.
Normally, yes. But chemo agents, like topoisomerase inhibitors, need the cell to be mechanically active. If a cancer cell is dormant, sleeping in the G0 phase, its DNA is packed tightly away.
So the chemo just flies right by it. It has no exposed target.
Exactly. But if the cell is running that M7S program, its DNA is unzipped, exposed, and incredibly vulnerable to structural damage.
Ah, I see. And the M13ER stress program just adds to that vulnerability.
Yep. If the protein factory is backing up, the cell is already triggering its own unfolded protein response. It's teetering right on the edge of apoptosis, or programmed cell death.
So the chemotherapy just gives it that final little biochemical push over the cliff.
Precisely. Now, the other 2 protective metaprograms tie back to that immune mimicry we talked about. M5 is the internal interferon signaling, and M6 is HLA expression.
Okay, HLA, that's human leukocyte antigen. Normally, professional immune cells use HLA to display like crushed up bits of viruses on their outer membrane, right?
Yeah, to flag down the wider immune system and say, hey, attack this.
So for a cancer cell to actively run the M6-HLA app, that's wild. It's essentially a burglar wearing a high-visibility neon vest that says, I stole this.
That's exactly what it's doing. It is literally placing its own mutated abnormal proteins in its front window, practically begging the immune system and the chemo to destroy it.
Wow. Okay, time for another quick recap. Bullet 1, inside the tumor, cells run specific programs. Bullet 2, cancer cells running the dividing, stressing, and immune-mimicking programs are the easiest to kill.
Because they expose themselves to the drugs.
Right. So, question for you listening. Knowing that active division makes cancer vulnerable, how might that change the way we think about the speed of tumor growth? Sometimes a fast-growing tumor might actually be easier to treat than a slow, dormant one.
It's totally counterintuitive, but absolutely true. But, even a highly vulnerable cell with a neon vest can survive if it's hiding in a reinforced bunker.
Oh, right. Because cancer cells don't live in a vacuum. They live in a neighborhood.
The tumor microenvironment, or the TME.
Right. Having established what the cancer cells are doing, we now have to ask, who are they hanging out with?
Exactly. And using that Xenium spatial data, the researchers mapped out 49 different TME cell states. They found these things called ecotypes, which are networks of cells that always hang out together, and spatial niches, which are the exact physical coordinates where they live.
Location, location, location. So what were the key neighborhoods they found?
Well, there's a really good one, ecotype 8, located in what they call niche N2. In this neighborhood, those vulnerable interferon-running cancer cells are physically surrounded by interferon-associated T cells, B cells, and macrophages.
So the burglar in the neon vest is literally standing in the middle of a heavily armed immune SWAT team.
Exactly. And the clinical data shows exactly what you'd expect. Tumors dominated by ecotype 8 have fantastic responses to chemotherapy. They get wiped out.
But I'm guessing they found a bad neighborhood too.
Oh, really bad one. Ecotype 3, located in niche N4. Here, the cancer cells are running hypoxia programs, meaning they're starving for oxygen, and they are clustered tight with a very specific cell called an angiogenesis macrophage.
Okay, angiogenesis. That's the biological process of building new blood vessels. So the tumor core is suffocating, and it recruits these corrupted macrophages to act as microscopic plumbers.
That's a great way to put it. They lay down new vascular pipes to feed the cancer. But the pipes they build are terrible. They're leaky, they're poorly structured, and they create massive interstitial fluid pressure inside the tumor.
Wait, fluid pressure, so it's a physical barrier?
Yes. That abnormal pressure gradient physically prevents the bulky chemotherapy molecules from even diffusing into the tissue.
That is incredible. The resistance isn't just chemical, it's architectural. If your cell lives in the niche N2 interferon district, it gets raided easily. But if it lives in the niche N4 hypoxia slums, those corrupted macrophages have literally built a pressurized bunker around it.
It totally changes how we have to view treatment resistance.
Okay, let's do our third recap. Bullet 1. Cells group into distinct ecological communities. Bullet 2. Some neighborhoods invite destruction, while others build physical, pressurized bunkers to block the chemo.
Right.
So think about this. If these are microscopic physical neighborhoods, what external factors, like, say, a patient's vascular health or systemic inflammation, what might change a neighborhood's pressure and make it suddenly responsive?
It forces us to see the tumor as a dynamic, reactive organ, not just a static lump of bad cells.
Okay, but let's bring this down to reality for a second. We've mapped the city, we know the gangs, we know the neighborhoods, but mapping a tumor at a 400,000 cell resolution using spatial transcriptomics, that's insanely expensive. Right?
Oh, prohibitively expensive.
Yeah.
You can't mandate that for every single breast cancer patient walking into a regional clinic. It takes immense computational power.
Right, so how do we actually use this to save lives right now?
Well, the researchers knew that bottleneck existed, and this is where the data science part of the paper is just brilliant. They didn't just build a map. They built a machine learning pipeline to find a clinical shortcut.
A shortcut? How?
They took all this incredibly dense, high-dimensional data and fed it into a feature selection algorithm. Specifically, they used penalized logistic regression.
Okay, translate that for us.
Basically, the algorithm mathematically shrinks the predictive weight of all the irrelevant or redundant genes down to 0. It throws out the noise until it finds the absolute minimum number of variables needed to predict whether a patient will achieve a complete response or be left with residual disease.
Wow, and what did it narrow it down to?
A concise 13-gene signature.
Only 13 genes out of thousands. And the beauty of a 13-gene signature is that you can test for it using that standard, cheap, old-school bulk RNA sequencing.
Exactly. You just run the biopsy through the standard blender, but now you know exactly which 13 molecules to look for in the smoothie.
That is so elegant, but how accurate is it? I mean, what are the performance metrics?
The model achieved an area under the curve, or AUC, of 0.84.
Okay, for those who might not know, in clinical statistics, an AUC of 0.5 is basically a coin flip, a random guess, and 1.0 is perfect prediction.
Right. So an AUC of 0.84 is a highly robust model. It brilliantly balances sensitivity and specificity so clinicians can actually trust it.
But, you know, a model trained on one specific data set will always look good on paper. Overfitting is like the classic trap in AI and computational biology.
Absolutely.
How do they prove this 13-gene signature actually works in the real world across different demographics?
They rigorously validated it against massive independent historical patient cohorts. We're talking gold standard data sets. They tested it on the ISPY2 trial data, brightness, metabric, and scan-B.
And it held up.
It held up across the board. The 13-gene model successfully predicted the risk of residual disease, and it correlated directly with overall patient survival across all those diverse groups.
That's huge. And I read that while they were running this predictive model, the algorithm actually surfaced a really surprising insight about the tumor hierarchy.
Yes. When it was calculating the predictive weights, the specific states of the macrophages were dominating the algorithm's decision-making process.
Wait, really? The macrophages were more predictive than the cancer cells themselves?
In many ways, yes, because the macrophages are the architects. Whether they're building those leaky blood vessels in niche N4 or recruiting T cells in niche N2, they dictate the physical reality of the tumor. The math essentially proved that the surrounding ecosystem is just as critical to survival as the actual cancer mutation.
It's all about the neighborhood. Now, the researchers also identified a backup plan, right? A therapeutic target hidden in all this data for the patients who do have those resistant basal-like ARC2 tumors.
They did. They found that across almost all the different archetypes, including the highly resistant ARC2, the cells heavily express a surface protein called TOP2.
Okay, TOP2, why is that important?
Because TOP2 sits right on the cellular membrane, and it rapidly internalizes whatever binds to it. It just swallows it whole.
Oh, I see where this is going.
Yeah. It is currently the prime target for a revolutionary class of therapies called antibody-drug conjugates, or ADCs.
Which are basically microscopic Trojan horses.
Exactly. You take a targeted antibody, you link it to a highly toxic chemotherapy payload, and you send it in.
The antibody binds to the TROP2 receptor on the ARC2 cell. The cell swallows the receptor, and the toxic payload detonates exclusively inside the cancer cell.
Right. It completely bypasses the spatial resistance of the microenvironment bunker because it's delivered straight to the target.
The clinical utility of this entire study just perfectly comes together right there. We have a biological map, a cheap diagnostic shortcut, and a targeted alternative therapy.
It's precision oncology at its finest.
So when you pull all these threads together, the 4 archetypes, the metaprogram apps, the spatial neighborhoods, and that machine learning model, it fundamentally changes the conversation in that clinic we envisioned at the start.
It really does. In the very near future, a doctor won't have to guess and just hope for the best.
Right. They can run a simple, inexpensive 13-gene bulk sequencing test on a biopsy. And they can look at patient A and say, your tumor is dominated by the ARC3 archetype. It lives in a highly vulnerable niche N2 neighborhood, and standard chemotherapy is going to clear this disease completely.
And for patient B, they look at the exact same readout and say, your tumor is running basal-like programs. It's built an impermeable vascular bunker. Standard chemo will only poison you without curing you. So we are immediately skipping the standard protocol and using a TOP2 targeted Trojan horse.
We replace a devastating systemic trial and error approach with actual foresight.
Exactly. It saves lives, and it saves people from unnecessary toxicity.
Which leaves us with a truly fascinating theoretical horizon to end on. Because if we know exactly how these neighborhoods operate, right?
Right.
And we know that cancer cells in the vulnerable ecotype 8 neighborhood mimic the immune system by upregulating HLA.
Right. They invite their own destruction.
What if we didn't just read the spatial map? What if we actively rewrote it?
Oh, terraforming the tumor microenvironment.
Exactly. Terraforming. It's the next logical frontier. If a patient presents with an ecotype 3 tumor, the fortified resistant bunker, could we theoretically use epigenetic modifiers or some targeted biological agent to artificially force those cancer cells to turn on their HLA metaprograms?
You force the burglar to put on the neon vest before you even administer the chemotherapy?
Yes. We actively engineer the tumor's physical neighborhood to guarantee its own destruction. Think about how that flips the entire concept of oncology on its head.
We wouldn't just be trying to poison the bad cells anymore. We'd be microscopic city planners.
Rewiring the infrastructure of the tumor to make it cure itself, that is a tantalizing thought for you to mull over until our next deep dive.